coming here. Thank you those who have been coming many times, regulars, those who are coming for the first time. chaiandwhy happens on the first Sunday, third Sunday, and if a month has a fifth Sunday, the fifth Sunday of the month. It's an outreach initiative of TIFR to take science out of the lab and bring you something. Now, one very important announcement before we start today's program. From next session, the mid-month chaiandwhy venue is shifting. Okay. 2 weeks from now, we will be at Nehru Science Center. The mid-month venue is shifting to Nehru Science Center, at least initially for a few months. Mid-August is National Space Week, and it is going to be the Space Week begins on Monday, but the program is going to start on Sunday. It's going to be the inaugural event of National Space Week at Nehru Science Center as well. We are going to start with a chaiandwhy. It's going to be on India's journey in space and getting beyond the Earth and things like this by Mayank Vahia. That's going to be the mid-month, but it's not at Ruparel College. It is at Nehru Science Center. From now on, mid-month venue is Nehru Science Center. Really important. Please make a note of this. Of course, we'll send you emails and everything else. Okay, with that, let's get to today's session. It's a pleasure to welcome all of you here. Thank you for coming again on a wet, rainy day. And we have with us Amitesh Anand from TIFR, who works on, as you can see, evolution. Maybe you should come one step thing because you are blocking it for the people here. Okay, so he actually works a lot with bacteria and antibiotics and how they develop resistance and how you can get evolution to happen quickly in the lab, so you can actually have a test bed for evolution in your lab, etc., which he will talk about. Amitesh starts his journey in Bihar, moves to BHU for his bachelor's, then he goes to Delhi, does a PhD there, goes to the University of San Diego, and then thankfully we grab him and get him to TIFR, where he's been with us for the last 5-ish years or so. 5-ish?
4.5.
4.5. 5.
Yeah, inching there, yes.
Inching to 5, yeah. So we are very happy that he's agreed to do a chaiandwhy, and over to him. And please ask questions. If you're watching us online on YouTube, remember the only way we can get your questions is via the chat. So please put your questions and comments in the chat. I'll monitor the YouTube, and if there are interesting questions, of course, we will interrupt him if it's important, or we'll take it at the end. So yeah, come in, join in. And let's get started.
Sure.
All yours.
Cool. Thanks.
Now you have to share screen.
Important, yeah. Am I good?
Yeah.
Cool. Good morning, and thank you so much. As Anna was saying,
You can move the talking also somewhere out.
is that cool? Okay, so this is my second good morning to all of you. Okay, I don't mind. Well, as Arnav said, thanks so much for braving the rain and being here. This is an iconic venue, and thanks Arnav and Ullas for giving me this opportunity to talk about something that we do in the lab and how does that contribute to human welfare and welfare of life on planet in general. And I'll try keeping my talk as easy to understand as possible because my 5-year-old son is here. So I would appreciate if he understands a bit of what I'm trying to say. So to get started, this term evolution, and it's a household term. You must have heard of this term sometime in your life, in your education or in your daily life. So what we are going to talk about today is evolution in multiple different contexts. It will work. It should work.
So you shared screen now.
Yeah. Okay. So what we are going to do is that we are going to keep this talk an engaging talk. It should not be I just doing all the talking. I would appreciate if you interrupt me, ask things, do the clap, and I have propped him there to. Okay, cool. So, to begin with, when you hear this term evolution, something might have came to your mind. You must have visualized something. So, what is that? Would any of you care to talk about what you visualized when you heard this term evolution? Or it's too early? I'll wait for you to get more engaged in it. So, your visualization, if I anticipate, will depend a lot on your academic background, your background in general, and at what stage of life you are. If you are too much into biology, if you're too much into science, then you will start visualizing evolution long back, all the way from Big Bang and whole evolution of first life on Earth, and then how those tiny life have diversified, evolved, resulting in formation of a sophisticated organism like us. But for majority of us, the concept of evolution starts from a little bit more visible organisms, like fish that we know of, we have seen it, and how these organisms started moving out of ocean and started colonizing land. And this is a very beautiful event. And what happened back in history when evolution of life was taking place is these fishes were trying to get out of the land. So one of the way for them to get out of the land was to be able to respire in air, using air. So, you know, fishes have gills. So they then started transforming into lung-like structure. And a fun fact is this guy over here is called lungfish. And the amazing part about lungfish is that, and this is one of the oldest fish available in any aquarium. It's in California Academy of Science Stenhart Aquarium. This lungfish is amazing in the sense that it has both gills and lungs. So it can respire in water as well as on land. And this is something called, in evolution, missing link or living fossil. Basically, you have something which is telling you that evolution is taking place. There is this smooth transition, there is this gradual transition of life from one form to another. And then this fish, through these lungfish kind of thing, started appearing on land in the form of something called amphibians. I'm again sure that you must have known of frogs, toads, these kind of things. And these creatures have to spend some time in water to complete their life cycle, or at least to produce their offsprings, but they can very happily live on land. Then came reptiles, very interesting set of organisms. Right now, we know more of lizards and all, but again, you must be knowing about dinosaurs and all. So very interesting set of life. And then from there, mammals and the whole human evolutions, everything. So this again is, if you are slightly more inclined toward biology and you are able to follow through the evolution of different organs, are able to relate and connect these things. For even wider audience, the concept of evolution is slightly more restricted to human evolution. Okay, and this is where we all, to a different extent, agree that we have evolved from monkeys, apes, these kind of things. So there has been very gradual evolution, and this whole Homo evolution has taken place. So apes are our ancestors, and through that we have evolved to what we are right now. And the fun fact is that we are still evolving. As fact, like Homo sapiens as a human, we are still evolving. Now the cool thing is that, and this example I was giving to my lab yesterday, is that this human evolution is helping dentists a lot. Any dentist here? Good. So it's helping the dentist a lot, because what has happened in human evolution, or particularly human civilization evolution, is that our food habit is changing. So we now cook food and eat slightly more sophisticated kind of food. So our dependency on our molars for chewing those food is reducing, because of which you don't need full set of molar. You don't need the something called wisdom tooth. And I'm sure many of you, not younger ones, but my age or older, including my wife, we have experienced this thing where this wisdom teeth does not always come in a very right orientation. And you visit dentist, not so much in India, but in US, if you visit dentist, even if you don't have any problem with your wisdom teeth, they'll ask you to go for extraction. It serves no purpose in our day-to-day life. Second, what is happening because of change in food habit, the space required for the development of this third molar, which is your wisdom teeth, is shrinking. Because of that, you don't have enough space for the proper growth of this particular set of teeth. That's why you have to ultimately get rid of this, and dentists are getting benefited because of that. There are many more examples of how humans are still evolving. The whole fact of we becoming lactose tolerant. So you must have heard of lactose intolerance. So lactose intolerance was the norm of life. Most organisms, yes. Right. So that exactly is, yes, it is a part of evolution, because gradually all of us will get rid of it. Right now that space is shrinking. Over the period of time, the cells or the progenitor cells required for the growth of those teeth will go away. It is not necessary. So that's why a large part of Indian population remain.
Can you repeat the question to the online audience?
Okay. So the question asked over here is that if these wisdom teeth is not doing any harm, why to extract them? Right. So first of all, I'm not a dentist, and I'm not suggesting you to go for the extraction. Okay. I'm not trying to popularize any business. Okay. Having said that, in most cases, this particular set of teeth does not come in right direction, right orientation. Okay, so that start hurting your set of other teeth. Okay, second thing is that because it's so back in your jawline, that cleaning that become an issue. And if cleaning become an issue, and if you're not very good with flossing, then you run the risk of harboring pathogens or microbes that will then start hurting other set of teeth also. So that's the reason why in US, if you go to the dentist, no matter what, they will simply ask you, get rid of it. Of course, business, but your help also. Okay, cool. So that's that. What I was saying is that.
But I think this is limited to the US because
evolution, yeah, sir.
When I was starting my career, I had a choice to join a group that was working in Antarctica.
Right.
It was very exciting.
Right.
Because after going there, when you come back, you don't find Midwest winters cold at all. But the condition was that you have to extract your wisdom.
Okay.
And I said, no, I'm not going to do it. Then when I went for a postdoc in Germany, they said, very good. Because even if the tooth is not useful, later on you might lose a tooth, and having a tooth next to it is good because it's easier to make a bridge.
That's
again a business.
That's a.
There was a no, unless there's something wrong with your tooth, we're not going to extract.
Fully agree. So again, I'm saying that there is no norm that everyone should get rid of it. Okay, it's just that it's serving no purpose, and evolution is shrinking the space. So it's highly likely that this tooth will not come in right orientation. Well, in general, they are more evolved in that side, but yeah. Well, in fact, the fact is that to second what you said is that between I and Manisha, my wife, I have more wisdom. I have both of my wisdom teeth. She lost both. Oh, the wisdom in that sense. Otherwise, she has more wisdom than I. Yes, please. Right, right. Yes, that's the course of evolution. Okay. So given we had set up a little bit of interesting discussion over here, so I'm going to cut short on the lactose intolerance part. Just that we were all lactose intolerant. Right. Right. So canines are So right now we have both set of food habits. So we still need canines, although for certain food preferences and certain kind of population, the dependency on canine is less. So of course you can survive without canines. But in general, human population, what you are talking about is more shorter period of evolution in human life. I'm talking about a very broad evolutionary scale. And the changes that we're talking about takes place in thousands and thousands of years. So if our food preference start becoming more and more non-canine dependent, then over the period of time, that will also happen. Yes.
Does the lack of oxygen for these food cause evolution? Because to my understanding, evolution works through a certain mutation which causes.
I got your question, and I'm sorry I'm interrupting you, but I've got your question. And I have full set of slides to explain what you're asking. Okay, cool. Now I'm going to skip the lactose intolerance part entirely. Just a disclaimer that we were all lactose intolerant at some point, and evolution has made us more and more lactose tolerant. Okay. So as said, for most of us, when we talk about evolution, it's more about human evolution all the way from apes to the modern human. Okay. And even more relatable is these kind of maps where in books we have read that initially this human evolution took place in Africa, and then from there it diversified to different part of the globe. And then you have Homo erectus, Neanderthals, and then Homo sapiens and all coming. And these are the kind of various human forms that you are more aware of. You visit museums and all, and you'll get to see all these things. We have tons of fossil evidences of talking about how these human forms have changed. There is something very interesting about this part of the structure of your face. Svante Pabo, who got Nobel Prize, I think 2 years ago or 3 years ago. So he has worked on human evolution a lot. So you'll be very excited to know that there was just one mutation that he was referring to. So there was just one mutation that had changed the shape from this to more flat like this thing, like bulging out of this. And this is our frontal cortex. This is for your intelligence. And that has made us more intelligent compared to our previous counterpart. Again, not going too much into it, but again, one of the things is that most of these things that I'm telling you, you have not seen firsthand. You have visited aquariums, you have visited various museums, you have seen these fossil evidences, or you have read it in books. Someone or the other has told you that this has happened, and they have said it in such a logical way that you tend to agree. Have you seen evolution happening? I don't think so, because as said, evolution happens in a very, very longer time scale. But before, sir, you said no to it, we are a generation which is amazing in a sense that we had this unfortunate fortune of seeing evolution happening right in front of us. That was just a couple of years during a COVID-19 pandemic. So COVID-19 pandemic is a clear example of how evolution happens and how evolution progresses. So there was first evolution of a virus that was earlier not infecting human, started to infect human. That happened somewhere in China, this Wuhan province. And then this virus had this pressure of escaping the immune response of host, and it also has the pressure of better transmission. So because of that, again, certain changes start to happen. And those changes are nothing but, as this gentleman was saying, this mutation. I'll go to that concept a little bit more in clarity. So these things started to happen, and you started seeing different variants. So you heard of alpha variant, beta variant, Omicron, et cetera, So this is nothing but evolution happening right in front of you. Cool. So this is evolution happening right in front of you. Now, what is happening here? So again, when you got to see these kind of tiny viruses, which is the COVID-19 virus, there is something over here. You see this flower kind of structure. This is called spike protein. Okay. Wherever you find jargon, feel free to ignore that. Okay. I'll ensure that my talk and our discussion is comprehensible without even going into the jargons. But being a scientist, I tend to use some jargons at a time. So please bear with me. So there's something called this a spike protein, and this spike protein is required for this virus to attach to your cell and then go inside. So basically infect you. And when the first COVID-19 virus came, this spike protein was not very well optimized for the infection. Now, during the course of evolution of this virus, they started better optimization of this spike protein so that they can infect you more and more. So that's what you see over here in this plot, is that December 19, this Wuhan one, it's a spike protein. So there's something called a furin cleavage site. Again, jargon, forget that. So this has started to optimize. And when you had the alpha one, it was more optimized. Then you had delta one, even more optimized. During Omicron, something very interesting happened, that you got two variants of it, one with better optimization, another with poorer optimization. And that's the reason why over a period of time, we started seeing decline in the infectivity. In fact, this virus has not gone anywhere. It's very much in our population. It's just that we have become adapted to live with it. So that's that. Now, all these things were happening because of some alphabetical changes in this virus. So these are some interesting alphabets that were changing in wild type to alpha to delta to Omicron. And these changes were the one making this virus become more transmissible and more able to infect. So now, what are these alphabetical changes? What are these typographical changes? This is nothing but something that we call as mutation. Now, how to understand mutation? So again, there are a couple of terms that you might have heard sometimes, something called genome, DNA, etc., etc. So every life form has inherent codes. So we all have something called code of life. So if my son has to look like me, because I and Manisha has passed on certain codes in him. Similarly, every life form does that. Every life form have a set of code that it transfers to its progeny so that they look more like it. And they, in some cases, they look almost very like identical to it. If you compare my 4-year picture with this guy, we look very similar. Sorry, Manisha, but we do look more similar. But that's the thing. Now, as with any book, the book of life that I'm referring to, the organismal book of life, is also based on certain alphabets. But unlike this 26 alphabets that we use in English literature, in this case, there are only 4 alphabets, A, C, G, T. And chemically speaking, these 4 alphabets are 4 different chemical bases. Again, jargon, skip it. These bases are something that constitute this molecule called DNA. And this DNA then condenses a lot to form something called chromosome. And you might have heard of that. We have certain set of chromosomes, and these are what is transferred to our offspring, half from father, half from mother. And that's how your offspring has a kind of a shared characters and all those things. So this is what is DNA. This is what is code of life. Okay. Now, how does this mutation come? As said, we have a code of life, and this code of life is like a book. And here I'm talking about an organism called Escherichia coli. This is it's a bacteria. It's a tiny organism. It's not visible to naked eye. You need a good quality microscope to be able to see them. This kind of tiny microorganism has a code of life which is written using 4.6 million characters. And just to give you a sense of how big this is, you only need 3.6 million characters to write the entire series of Lord of the Rings. You need about 3.2 million characters to write this beautiful book, War and Peace. And this tiny microorganism has its code written using 4.6 million characters. Now, if I just, yes. Characters are something that comes from this code of life. The phenotypic manifestation, so tallness, fairness, even intelligence, risk of catching some sort of infection. These, I would say, trait or characters. And the basis of this is inherent in your code of life. Right? Well, you can consider these bases also as character. Oh, these characters, yeah. These characters are alphabetical characters. I thought you were talking about the higher level character. So these characters allow, so the way in English literature, you use 26 alphabets to write something. Similarly, in Book of Life, you need these 4 characters to write the entire Code of Life. Okay. Now, this is the time where I would now want you to engage more with me, is if I give you this entire set of books and I ask you to write it. Okay. I'll ask my millennial colleagues to write it and my Gen Zs to type it. What do you think, how much time you will take to type this or write this entire book, end to end? Any of you think we'll be able to do it in a day? No, right? Even if I ask you to scan it, forget typing or writing, just take a scanner, just keep on scanning it. It will still take, sorry, I heard some hours. 3 years. 3 years. Sir, you seem to be a bit on, yeah, you need to fasten up. It will, but certainly it will take a lot more time. Okay. Now, the thing is this guy does not have that privilege. This guy divides every 20 minutes, which means that it has to rewrite this entire thing in less than 20 minutes if it has to pass on the information to its progeny. That's the pace with which they write their codes. If you write this, even if you are very diligent, and if you take one of the character about you in the sense saying 3 years is that you want to write it in a perfect way. You want to take enough time so that you don't make mistakes. But I can assure you that even if you take 3 years, you will end up making some typographical errors. Okay, these typographical errors, in case of Code of Life, is nothing but mutation. Okay, and mutations are of all kinds. Not every mutation result in some changes. Okay, the example over here is that if I read somewhere written, I believe in evolution, and if I want to rewrite it, and I made a mistake that I missed this last E over here, I write it, and then if you have to read, you'll probably still be kind to me and read it as I believe in evolution, right? So this change is not as manifested as the second example over here. So if I read somewhere written, I am native to India, and I make a small similar error that I miss this T over here, it becomes I am naive to India. Now it has very different meaning. Similarly, in case of Book of Life, there are tons of mutation. Some are manifested in certain changes, others are not. And this is important because if every mutation start to reflect in some character changes, then we will start changing every time we give rise to another offspring. So that's that. So these are the changes that becomes that brings in heterogeneity in life, and then that becomes raw material for evolution to take place. Now, during evolution, what happens is, just give me 30 seconds and I'll come to you. During evolution, what happens is that there is something called selection pressure. So basically, selection pressure is nothing but something that requires you to acquire a new character. Like when human were started to keep cattle around and started using milk for consumption beyond their early phase of life, then the pressure was to be able to digest it. So then the human who had the mutation in this enzyme called lactase becomes selected. So there is natural selection to select a certain trait out of existing pool of heterogeneity. So that's the natural selection, and natural selections operate on this heterogeneity that comes because of typographical error or the mutation, and then you have a new life form coming. Yes.
For example, the mutation happens in protein. It's usually expected that the mutation will happen.
Right.
And these are like or in terms of mutation. And what decides whether this mutation will continue on for another person?
Right. So thank you. That's what I was about to do. I learned it now. So the question is that what decides this evolution, this mutation to then pass on to new generation or to be selected or not, and what decides some mutation to reflect in a change versus not, right? So one, now she's going a bit on a textbook, so I'll have to use some jargons, okay? So there is something called, so I talked about those 4 characters which constitute this entire code of life. Out of those 4 characters, 3 characters together form something called a codon, which is equivalent to a word in book. Now, not every change in this 3 codon result in change of protein, okay? There are several words carrying same meaning, happy, glad. Similarly, in codon, there are several codons which carry message for the same amino acid of a protein. So not every change will result in change in the protein composition. First part. Second part, what decides whether if there is a change which is actually resulting in change of the protein, whether that will be passed on or not. That's where selection pressure comes. If the change has made the protein worse, it will not be passed on to new generation. But if the natural selection is favoring that change, then it will be passed on. Okay, cool. So it depends on which organism we are talking about. Okay, I work with bacteria. So have you heard the term bacteria? These are tiny, like this is even a lot for bacteria. It's like really, I have few plates I'll show you after the talk. So when they divide, so what happens with bacteria is that they grow to a certain size, and then something happens and they decide that now we have to become 2. So they divide. But before they divide, they rewrite that entire book of life. And after division, both cells will have 1 copy of the book each. So this is for bacteria. And for human, it's based on this parent to offspring, next offspring, generational changes. Sorry? They have to do a lot of protein synthesis because writing this book of life, like in our case, we require ink, pen, and all to write. Similarly, in case of cell, they need lots of different enzymes, which are nothing but proteins, to be able to write these things. So they need protein synthesis. Protein to code. Okay. Right? Yeah. Again, it seems you're quite aware of the things that you're talking about, so I'll use some jargons again. There is a process called replication. Okay, replication is nothing but rewriting or reprinting that book. Okay, so what happens is that there is a cell that has 1 code written, and now this cell has to divide in 2, and it has to pass on that information to the next one. So there comes the entire machinery of enzymes and various other molecules required for rewriting it. Okay, then it got rewritten, transferred, and the code that you write is also responsible for the production of the proteins which are responsible for the rewriting of these codes. So that becomes the process called transcription, translation, Latin mass, and I'll spend a lot of time with you. Okay, so then replication, transcription, and then translation. So there are a lot of these things, something called central dogma and all those things. I will ensure to spend a lot of time with you, and I'll tell you more about it. Yes. Yes, yes, there are.
That's the way just the way all these diseases get passed on.
They do. Well, there are 2 elements to your question. One is, so the question is whether different parts of the genome has different susceptibility to mutation, and what dictates whether that mutation will be transferred to the next generation or not, right? So for the first part, answer is yes. Different genomes have different susceptibility depending upon what kind of composition of these alphabets exist. There are certain segments of genome where there is something called repeat elements. So these alphabets repeat a lot, and they are more prone to mutational changes. Okay, But this susceptibility is not something that dictates whether that will be transferred or not. Transferred or not, or selected or not, depends on selection pressure. Whether the changed protein or the changed phenotype, changed character, not the alphabetical character, okay, whether that is providing them an advantageous change, making them more adapted to the environment. That dictates what will get transferred or not. Okay?
You can proceed with the talk. After the break, I will.
Sure. So that brings me to episode 2. So I have recently finished Dhurandhar, so I have also followed that episode wala style. It seems these days this thing is in fad. Yeah, thank you. Yeah, for the first episode. Now, the second episode, this will connect more to what we do in our laboratory. Okay, I talked about this entire long length of evolutionary time scale, and this chapter is going to talk about one particular period of evolution. This is about 2.4 to 2.6 billion years ago, what happened on Earth. Okay, it's a huge time. Okay, it's almost half midway the life of Earth. So during that time, something catastrophic happened, something that we call as mass extinction. And if I say mass extinction, you will start visualizing something of this kind, right? Vedant, you know this, right? So you know of this dinosaurs. You believe in dinosaurs, right? Yeah, they existed on Earth. Okay, that's not just a character for they to watch on TV. We have proper fossil evidence and everything. They existed on Earth, but now they are no longer there. This kind of huge creature not existing is almost difficult to imagine. But something happened on Earth during that period of time. There are lots of theories. One theory is in his book that there was a huge war of underpants, but that's not the right one. One of the most acceptable theory is that some extraterrestrial object hit our Earth, and that resulted in a huge geochemical change, resulted in global cooling or the cooling of the Earth, and that's most plausible reason for the extinction of dinosaurs. Now, the thing is, this is one event that happened during this Cretaceous period of Earth, which we are more familiar with. And during this period, it's not only dinosaurs that got extinct. In fact, according to some estimates, some 75% of the life form that existed on Earth during that time got wiped out. Okay, so it's a huge extinction event. But the event that I'm going to talk about, this event is nothing compared to that. So that is something called Great Oxidation Event. And that actually changed Earth to look something like this. Okay, so what really happened? Why, during that period of time, Earth turned so hostile? So what happened is, there was this evolution of a set of organisms that we call as cyanobacteria. What they did is that there was this abundant sunlight. So we all need energy, right? And one of the purpose of the life is to conserve energy to be able to do whatever we are doing. So abundant source of energy is sunlight. So some of these organisms figured how to utilize sunlight or how to conserve sunlight by developing something called a pigment that was able to capture the light or energy from sun, and then using CO2 and water, they were able to form something called carbohydrate. This is a process we generally refer to as photosynthesis. The outcome of this process is oxygen. So this is for the first time Earth started to receive oxygen. Before that, either there was no oxygen on Earth or there was tiny quantity of oxygen on Earth. Okay. Now, if today I tell you that oxygen is a poisonous gas, some of you will be like, this guy is talking nonsense, right? Because oxygen is, that's the word you suggested me yesterday. Yeah. Because oxygen is almost synonymous to life. Okay. But even to us, if there is a deviation of oxygen in our environment, we also will face the burden. And I'm talking about a time when organisms that were existing there had never seen this molecule. So they were just not prepared to deal with this molecule. And it is a reactive molecule, and it takes various forms which become even more reactive, something called reactive oxygen species. They are damaging, they kill the cell. So this toxic molecule started coming in environment to organisms that were just not prepared to deal with it. Now, the second thing that happened because of this oxygen coming in environment is that oxygen started to react with a particular element called iron. Iron you must have heard of. It's a very essential element for life, and it has been essential throughout the evolution of life for a long time. Now, iron is a very difficult molecule for organisms to acquire in modern environment. This is because oxygen converts iron from one particular form to another particular form. Now, a little bit chemically speaking, what happens is that oxygen oxidizes ferrous to ferric, okay, because of which this becomes insoluble. So the excess of iron becomes very difficult. So that was another challenge because there was free supply of iron in the environment, but then comes oxygen, it becomes insoluble. Now the essential element that you need is depleting. The third thing was something that, again, we are well aware of, something called greenhouse gases, global warming, all these things that we hear of. So when you hear greenhouse gas or global warming, many of you will start picturizing a negative thing about it, okay? But greenhouse gases, global warming, these are very important things for us. These are the one that maintains the temperature of Earth that's required for us to survive. Now, what happened during this time is that when oxygen started to come in the environment, oxygen started to react with a molecule called methane. And methane started to convert to another molecule called carbon dioxide. Now, the difference between methane and carbon dioxide, other than the elemental composition, is that carbon dioxide is poor greenhouse gas compared to methane. So what happened is that Earth started to lose the thermal blanket. So you have rise of toxic gas, you have depletion of essential element, and then you have global cooling. These three things together led to the most catastrophic mass extinction event on Earth. And again, there's an estimate that during this period of time, 99% of the life that existed were wiped out. Some existed, yes. How does oxygen become? Oxygen is always poisonous. You have to learn to deal with it. We have learned to deal with it. I'll tell you how after the talk, okay? So as said, during this period of time, there was a huge extinction event happening. But as with everything, and because we are here, we are existing on Earth, which clearly says that life found a way, life always finds a way. So a lot of changes happen, lots of adaptive changes happen, lot of typographical error, meaningful typographical error happened, and there was this evolution taking place. One of the changes that happened during that period of time, which we are interested a lot in, is diversification of a particular chemical molecule called quinone. These are some quinone that some of you will be aware of. We take it as supplements also. There's a ubiquinone, CoQ10. This is supposed to help you with your better cardiac health. It's also supposed to be anti-aging and all those kind of things. And these molecules being anti-aging or helping you with cardiac health is because they support your energy generation. They support something called electron transport system. They prevent formation of reactive radicals, which otherwise will result in aging and all those kind of things. So what happened during this period of time, that there was a very subtle change in a molecule which is called as naphthoquinone. This is an ancient quinone, and this is a modern quinone. So this is the quinone that evolved during the period of great oxidation event. And I'm sure that if you just look at these two, you don't really need to know chemistry for it. If you just look at it, you will say that these are very similar, other than this small part over here. And this subtle change in the chemical structure that took place during that time had a transformative change on life that were adapted later to deal with oxygen. Now, we wanted to figure out why this new quinone type came. Okay, so for that, these were the bacteria that were living in ancient environment using the ancient quinone type. Then came during great oxidation event certain set of bacteria that has biosynthetic ability for this modern quinone, and they are more adaptive in aerobic environment. And then of course you have some bacteria which are even more advanced. They only have the modern quinone type. So we wanted to figure out why ubiquinone came and how does this help in survival in modern environment. So for that, what we wanted was to recreate everything that might have happened on Earth some 2.4, 2.5 billion years ago. And I like this person a lot, Richard Feynman. Some of you, if you know him very well, if not, just follow his writings and all. He writes beautifully. One of his quote is, what I cannot create, I do not understand. And we take this advice to our heart and we follow this. Like when we have to understand a scientific problem, we try recreating that entire problem to understand it. So one of the way we are able to do that is on this row a lot, and so some of them are sitting. We are a bunch of people who have this ability to play around with the code of life, particularly bacterial code of life. So we can actually take a bacteria and we can change their codes in laboratory. So what we were able to do is that we took a bacteria that had biosynthetic ability for the modern quinone, we changed its code, and we deprived it with the modern quinone. So now this bacteria has to survive with the ancient quinone. Then what we did is that we took this one and we evolved it in the modern environment, which is very easy to simulate in the laboratory because we are in modern environment. So what happened is that, oh, I missed one slide. So when we say that we did this evolution in laboratory, what it means is that we made that particular organism and we kept on subculturing it. The same way you have generation after generation after generation. So we did this evolution for almost 1,000 generations. And if you want to appreciate the scale of this evolution, just think of you, your parents, your grandparents, your great-grandparents constituting 4 generations. Here we are talking about your 1,000th generation. Just think of where you will have to go back in time to go back to where our evolution starts from. So this is the length of evolution that we do. Of course, we are working with bacteria. They divide way faster than a human or any other life form. We are able to do these things in a relatively shorter period of time. So we did this kind of evolution. And then what we observed over here is that this is the bacteria that has modern quinone type. This one is only having ancient quinone type. If you give these 2 bacteria same amount of time to divide or to build its population, what happens is that this guy is able to build a larger population compared to the one that is solely relying on the ancient quinone type. Now we wanted to figure out why. So now bear with me a little bit of chemistry. So what happens is that you have this molecule, which is the modern one. This molecule, when it takes electron, converts to something called an aromatic molecule. Okay. Simply put, a very stable molecule. Because this is a very stable molecule, it will not go back to its previous form easily. So the electron that goes into this molecule will not come out of the molecule that easily. Compared to that, if you take this ancient quinone type, which is already aromatic, in this case, the stability difference between these two is not huge. It's not large enough to prevent this molecule to go back to its oxidized form, in which form they will be able to really leak this electron, and this electron forms a very reactive molecule or a toxic molecule called reactive oxygen species, and they result in killing of the cells. So what happens over here is that because these guys are exposed to these kind of reactive oxygen species, they are not able to build their population to the extent as the ones that has modern quinone. So bringing modern quinone made these organisms more tolerant to a molecule that came in post-great oxidation event, the toxic molecule that came post-great oxidation event. So this is what we learned from here. Now, in 3rd episode, what I'm going to tell you very quickly is about, I convinced to you that microbes can evolve, they can change their form. So if they can evolve, if can get new characters, they can also get resistance. Okay. Simply put, bacteria fight back. And here we are talking about bacteria which are bad, pathogenic bacteria. So we all are aware of this concept of antibiotics, that whenever there is this bacterial infection, and if your immune system is not being able to take care of it, you take antibiotics. And there was this discovery of antibiotic that took place around 1920 by Alexander Fleming, this whole story of penicillin and all. So this was one of the most amazing advancement in medicine to get antibiotic. And since then, we have developed and discovered many different antibiotics. But these microbes have also not been sitting idle. They have also been parallelly trying ways to deal with these antibiotics. So when we have been getting new antibiotics, they have been getting new ways to deal with those antibiotics. That's why many of the antibiotics that we take today are ineffective in treating the disease. Simply said, these strains or these bacterial pathogens are called antibiotic-resistant pathogens. And it is becoming a global crisis to the level that there is this disease called tuberculosis, quite prevalent in India. Statistically, in this room, every third person will have this infection. It will not reflect in any symptom because if your immune system is good, if you're taking good care of your health, then your immune system is keeping this bacteria in check. So there's no symptom coming, but you might have it in your body. And God forbid, if your immune system goes down for some reason, then they will reactivate and start showing the disease manifestation. Okay. So that's the impact of antibiotic resistance. And this particular bacteria, tuberculosis, has taken all sorts of form. Initially, it was drug resistant. So it was resistant to one drug, one antibiotic. Then it became multi-drug resistant, something called MDR. Then it became XDR, extremely drug resistant. Now we are talking about a new term called TDR, total drug resistance. So this problem has become a nightmare for medicine and for human in general. The ability to be, like when the first antibiotic came, everyone thought that infectious diseases are gone. We have this magic molecule, will take care of all the infection. Nothing will be like, in fact, some of scientists also started saying that, okay, infectious disease is something well taken care of. Later to learn that it was nowhere close to be even taken care of. So that's the thing. Now, even Fleming, the person who discovered penicillin, in his Nobel banquet lecture, has talked about, yes. How does that fight back work? Okay, I'll give you probably one quick example, and then there's a person sitting right next to you, Stuti. Grab her, she'll tell you tons of different mechanisms. Okay, one thing is that for this antibiotic to kill the cell, they have to get inside the cell. Okay, for them to get inside the cell, they have to use certain transporters. Okay, or even simple example is that these antibiotics target something in the cell to be able to kill the cell. What they do is that something called target modification. Okay, so whatever thing that this antibiotic was targeting, they modify that so that the antibiotic is no longer being able to target that. And they also play around with the route of entry. Either they block the entry or they increase the efflux, like they start kicking out the antibiotics from the cell so that it is not able to kill you. There are a whole set of different approaches by which these microbes are fighting back. They know a lot. Yeah. How can it's a sorry? Oh, please let him ask. Yes, he's asking beautiful questions. Please do. Quickly. Oh, no. How are you doing with time? Ah, good.
One also get killed, and one who are able to withstand the antibiotic will survive and will duplicate more.
Right. So that's the buildup of resistant population. You're right that once they acquire this trait that they become resistant, they are more selected. And that's why once you have this resistant bacteria, then the problem of resistant infection of resistant bacteria become more prevalent. Your question was different. Your question was, how does this happen? And you were saying that they don't have brain. Of course, they don't have nervous system and all those kind of things. They are a lot more sophisticated than what we think of microbes as. That can be one full talk. Okay, I'll give you some primer of it maybe in the next session, and I'll see if Arnav invites me again if I do a good job today. Then I'll talk about the brain part of bacteria. Okay, so what I was saying is that this guy Alexander Fleming, who actually discovered this antibiotic, he himself in his lecture way back wrote this thing. He's talking about an individual. He's just giving a very simplistic example that why to follow your physician's advice. When your physician is saying to take some antibiotic, this much dose of antibiotic for these many days, please follow that. Okay. Many times what happens is that it happens with me also, is that we start seeing symptom going down and we are like, okay, we are done, we are treated, let's no more consume that antibiotic. Please don't do that. Then you are actually providing these microbes a chance to evolve resistance. So whatever your physician is recommending you to do, please do, as long as you're visiting good physicians. Okay. So what he's saying over here is that while he has discovered penicillin, he's also thinking of a possibility that down the line, there will be bacteria that will be resistant to penicillin. Okay. And that's why he's saying that if you use penicillin, use enough. Okay. Don't let your enemy get a chance to learn to fight back. Kill them all. Here, bacteria, okay? Bacteria. Yeah. So what we do in the lab, from your side, you can do your bit by taking these antibiotics properly, by not letting this microbe get a chance to evolve resistance. That way you can help us. But we cannot solely pass on the blame saying that you are not taking the antibiotics properly and that's why the resistance is there. We have to deal with it. We have to figure out a way to fight back. So what we do is something called staying ahead in the game. Yes, still. So HGT, what's he saying is horizontal gene transfer. So that's the explanation, the question. Right. Right.
Or if, for example, if another person, like, we stop, like, for example,
So if you fail in taking your antibiotics properly, you are not only exposing yourself to the risk, you are exposing her, him, I, everyone. And this goes back to what he was alluding to, is if you have once this resistant strain coming, resistant bacteria coming, so this resistant bacteria comes because of something happened in the change of something called gene. And now these genes can transfer from one bacterial species to another bacterial species, one strain to another strain. So once you have one resistant bacteria that came, or that can provide opportunity for other bacteria also to become resistant. Right.
See, suddenly, like, I'm giving an example. If person A stops taking antibiotics and he or she hasn't been exposed to any bacteria properly, and then
Why are you taking antibiotics?
Like, I'm giving an example. This person hasn't taken antibiotics because they have never been having any infection.
Then why are you taking antibiotics?
No.
Okay, sorry.
Then they get an infection and they take antibiotics. So will they start working properly? Will it be working properly on this specific strain because it hasn't been exposed yet? Or will that strain, like, still have chances of having that same activity?
Yes. Quick answer is yes. You cannot take antibiotics in anticipation and be able to deal with it later. That's not how it works. That is the job of vaccines. Okay? It's slightly different. Over here, what happens is that these antibiotics are chemical molecules that directly goes and stop that pathogen. In case of vaccine, vaccines train you, your body, to fight back. They do not provide you some molecule to fight with the bacteria. They train you to fight better. And that's why I'm not sure I got your question very clearly. No. Vaccines and antibiotics are very different thing, okay? In fact, in many of the vaccine, you take the actual bacteria, the attenuated bacteria, the one that, sorry? Exactly. So that way what happens is that your immune system has already seen the enemy. So your immune system already know, okay, if this kind of enemy comes, then I have to fight back in certain way.
I thought vaccines only train your immune system to recognize the guy if it comes again.
Recognize and fight back.
The fighting is done by the body. I mean,
Yeah.
Does the vaccine teach you how to fight?
Yes, 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 yes. It teaches you what kind of antibodies you have to make when you have this kind of infection. Okay. Mostly those are replica of that. The antibodies replica of, sorry, what? No, they are not. That's again a beautiful question for a longer discussion. Okay, let me not delve into that right now, but the answer is that no, these are not replica of the bacteria, and they are not replica of any sort of antibiotic. These two are very different ways of dealing with it. Okay, yeah. So what I was saying is that what we try to do in the lab, using our ability to play around with the genetic makeup of bacteria and being able to evolve these bacteria in the laboratory, what we do is we try to stay ahead in the game. So before an antibiotic is introduced in market or be part of clinical practices, what we try to do is that we take that antibiotic in lab, we expose the pathogen to that antibiotic for several generations. It can be hundreds of generations, thousands of generations, till we see that this particular bacterial strain has become resistant to it. Okay. Now, we also, while we can rewrite codes, we can also read the codes by something called genome sequencing. So we can actually go back and read the entire change that has happened in that genome or in that code of life and learn what enabled this bacteria to become resistant. This way, we learn that if target A is being targeted, what B change takes place to make them resistant. And if you know this, then the chances of coming up with rational combination therapy is higher. The effective combination therapy is higher, because now what you can do is that you can target A, and you can block something that can help it overcome the defect of A. Yes. Right. Correct. Right. Right. Yes. In two ways. As you said, the context of human body is not always replicated in the kind of tubes that I'm talking about here. Having said that, we are trying to bring it more and more context. In fact, globally, there are lots of labs that have bring in a very close proxy of human-like state in labs. In my lab, there's this guy called Amartya. He is trying to develop some of that sort of approaches. But even the tube-based evolution that we do, it already allows you to figure out what are the pathways that can be compensatory, even without knowing the other context. If you know the other context, your information will be more resolved, you will be more impactful in coming up with the therapy. But this already helps you. Okay, so this is that. So what I said is that we try staying ahead in the game before the resistant strain appear in our life. We try learning about that in the laboratory, so that the evolution of those resistant strain in our daily life or in our population can be prevented. That's the hope, and that's the attempt that we put in our laboratory. With that, I am coming almost close to the end of my talk. I certainly want to acknowledge this person who has more wisdom than me without having wisdom teeth. And this guy is showing full set of teeth, my son sitting over here. And this is a brilliant set of colleagues I have in my laboratory. Some of them are sitting here. Grab them, they know more than me about these things, so they will tell you a lot more about these things. And of course, what we do in the lab requires money. And the money comes from various different sources in my case. And yes, if you like our work, and if you find our work relevant, and if you can just learn more about our work, and if you want, you can also support our work. We already have in audience some who are already supporting our work. So with that, I will not leave you without telling you that microbes are not villains. There are some there are some that are pathogens, but a large variety of microbes are actually not pathogens. They are very much part of our existence. In fact, half of us are humans, rest half is microbes. Okay, and another couple of weeks or a month, Stuti will take the stage to talk about something called microbiome, and she will tell you about how these microbes are amazingly significant for us. They play immense role in the health of your heart, being able to tolerate and not develop cancer, respiratory disease, and all sorts of things. So these microbes are very, very beneficial for us in our body. And these microbes are also required for the production of various sorts of metabolites. In fact, the antibiotic itself is produced using several bacteria. And for that matter, a lot of valuable chemical metabolites are produced using these microbes. So as said, as a microbiologist, I do not want to leave you with this negative thought that microbes are villains. Some of them are. Stay protected from them. Take your antibiotics as per the prescription of your physician properly. With that, thank you so much. More questions?
Okay. Just remember that you get a chance to ask questions after the chai.
Oh, sorry.
Hang on.
If you just allow me 1 quick minute.
Yeah, go ahead.
I have some props, okay, which I completely forgot to show you. I can elaborate more in the next session where you can come and see these things. So this is something called culture plate where we grow microbes, okay. This particular culture plate is almost equivalent of your healthy body, okay. So you won't see anything growing over here. It's a plain culture plate.
Stop share so people can see this, because otherwise you're a very small window on online people. Online people won't be able to see this unless you stop your share. I think it's okay. You can do that.
Okay. Okay. So as said, this is equivalent to your healthy body where there is no infection. Okay. Now, this is unfortunately representation of your body when you have a bacterial infection. Okay. And you see these tiny things growing over here. These are actual bacteria. Okay. As said, these are microscopic. You cannot see them, but you cannot see a grain of sand. But if you put a heap of sand grains, you will be able to see that. That's what you're seeing over here. These are like thousands of millions of bacteria coming together, and that's why you're able to see them. So this is a state of infected human body. Okay. This is the beauty of what Alexander Fleming did. So this is a plate where we have spread these pathogens or these bacteria, but we have added antibiotic. In this case, the antibiotic is something called ciprofloxacin. And now you don't see these microbes growing over here. This is what happens if you take right antibiotic in right way. If you don't follow your medical practitioner's guidelines, if you slack, then you end up with something like this. So this is a state of your body where you have infection. You have taken antibiotic, but you have not taken it properly. Now, this bacteria is slightly different from the one that I showed. This is the resistant one. So again, antibiotics, careful. Be careful with their intake. Be also careful with their disposal. So that's that. Now I'm
okay. So what we'll do is we'll take the online questions first, then we'll break for chai. We'll stop the live stream, and you can come back and ask him as many questions as you want. Okay. So one question is, can you please explain the bacterial evolution related to quinone again?
Okay. Yeah.
Come twice saying, can you please take the quinone question again?
Okay. I'm talking more to the online audience over here, so I will not be looking at you. Please bear with me for that. So the quinone evolution that I was talking about is diversification of a particular kind of chemical called respiratory quinones. And these quinones are part of our energy generation process called electron transport system. So in previous microorganisms, or the ancient ones, we had something called naphthoquinone, and they were not very good with holding on to electron stably. Because of their difference in some chemical structure, they were not very stable. And as said, they were already aromatic. So when they take electron and reduce, these two forms do not have a large stability gap. So these two can go back to each other very easily, resulting in a leak of electron, which becomes toxic to the cell. But the quinone evolution I talked about is where this particular ancient quinone changed slightly. And when it changed slightly, it formed a molecule when it reduces, become very stable. So it goes higher on its stability scale. So the chance of this to now auto-oxidize and come back to its oxidized form is less, because of which this electron that leaks is minimized, and because of which the toxicity is less and cell killing is less. So this was the evolution that happened during great oxidation event due to or in response to oxygen coming in environment.
Okay, there are 3 questions on antibiotic resistance and bacteria developing resistance. I'm going to put them together.
Sure.
You answer them in whichever order you want. So one is, how do bacteria get mutated over generations, establishing resistance over antibiotics? Can you explain this again? Then, is it not that in the lab, when you create a mutation for antibiotic resistant cell, it's a gain of function mutation experiment? Is it really allowed to do so after the pandemic? And I understand this will be Darwinism versus Lamarckism, but does the bacteria evolve to gain resistance when exposed to the antibiotic, or is the resistance always present in them irrespective of antibiotic?
All 3 beautiful questions. First, about mutation. So mutation does not arise in response to antibiotic. Mutation is a spontaneous process. These changes keep taking place. And this happens because of, so as I was giving the example of rewriting a book, you have the chances of making typographical error. Similarly, within the cells also, they have the chances of making typographical error. Their rate of error making is way low compared to what we do. As said, they rewrite this 4.6 million characters in a matter of 20 minutes or less, and they don't make a single mistake in that. When they rewrite this book 1,000 times, then chance is that one of the character will be written wrong. That's the beauty of these machinery in being able to precisely write the codes. But that one change out of 1,000, when you look at the bacterial population, becomes quite large. So this mutation happens, and this mutation happens randomly. So whenever you take a population of bacteria, you always have some sort of heterogeneity. No 2 cells are identical to each other at the level of their genome or genetic makeup. Now, when you administer antibiotic, when you put that selection pressure, then the one that has a beneficial change in its genome gets selected, and its population builds over time, and that's how you see resistant strain coming. Coming to the question whether this is allowed in the laboratory or not, what you ask is a very relevant question. And we already know that for the COVID-19, we are still dealing with this debate whether this was a man-made or natural, and jury is still out. I'm not taking favor of one or the other. We have not done proper investigation, so we still don't know whether it's a lab leak or it was a natural. There are lots of evidences suggesting that it's likely to be natural evolution, but the way this investigation has been conducted is also a bit fishy. So as said, we don't know what has happened during that time. What we do in laboratory is do it very safely and do it to the level that is already existing in clinics. So we are only allowed to recreate what is happening in outside environment. We are not allowed to create something new and have the risk of they running amok, creating havoc in our general life. There are lots of regulations and all, and every project that we do in the lab, or for that matter, any scientist do in their laboratory, has to be properly examined by a committee, national committee in our case, and every country has their own committee, and they look at what are we doing. They ask all sorts of questions. What are you creating? How safe it will be? How will you dispose it? And all those kind of things. So that's that. And the third question was about...
I think you've answered all of them.
Amazing.
There's one on the COVID thing. The COVID virus evolved to Omicron, but you said that evolution takes many years. So has the COVID virus evolved or has it changed?
Okay, so when I say evolution takes many years, I was referring to human evolution, okay? Because our rate of multiplication is way slower compared to these tiny organisms like bacteria and viruses. So the length of time or the time scale for evolution depends on which organism we are talking about. Because some of these organisms are very fast dividing organism. So the absolute length of time for them will be way smaller compared to higher organisms.
Okay, given that the chai outside is getting cold, I am going to sort of think those questions that keep coming. This one's on vaccines, and I think we should look at it in a talk on vaccines, honestly.
Sure.
I'm going to end with a funny one.
Okay.
You said microbes are also healthy and good.
Yeah.
Where do I get them?
Where do you get them?
Where do you get a dose of healthy microbes?
You're always taking that. You're eating curd. You're eating curd. You are consuming healthy microbes. You are taking probiotics. You are consuming healthy microbes. You are consuming dosa, fermented foods. You are consuming healthy microbes. Yeah.
Okay. On that note, let's thank him. Thank you, online audience.
Thank you.
Online audience, remember how this online is online from wherever we are. But physically, we will be at Nehru Science Center for the next mid-month episode. Right now, audience here, please have chai and come back. We have 3/4 of an hour for questions. We can continue with that. Have some chai and come back quick.
Sure. Thank you so much.